In the last video, we have described about the male reproductive structure in flower. And you, those missed that video, just go to the description box. I have put the link there. Just go and check the video. Today we will describe about the female reproductive structure in flower. We all know that the female part of any flower is known as gynoecium. And gynoeciums are known to bear a structure known as carpel. So this is a carpel, a typical carpel. If any gynoecium bear such kind of single carpel, then such gynoecium will be referred as monocarpellary. And if the gynoecium of any flower bear two carpels like this, same to same, then such gynoecium will be referred as bicarpellary. And if more than two carpels are present in any flower, then such gynoecium will be referred as multicarpellary. But today we will continue our discussion with such kind of monocarpellary gynoecium. Imagine there is a single carpel in the gynoecium and this is how it looks like. Let's find out its main parts. Typically, a carpel consists of three major parts. At the top, this structure is known as stigma. This is the site on which the pollen grains land. Then a long slender tube-like structure is present which is known as style, and at the base this solen structure is known as ovary. So in the carpel, this structure ovary is very important because it houses the female reproductive cell, that is female gamete. We will know it later. This structure, these are known as the ovules. So these structures are the ovule, and ovules are known as macrosporangium. That means these structures are known to house the female gametes. Okay, so there are 6 ovules are present in this diagram. It may be more than 6 or it may be less than 6, but I have drawn 6 ovule in this diagram. And this wall on which the ovules are stocked are known as placenta. Placenta. This is the placental wall of the ovary, and this free space is known as locule. So this is the structural details of any carpel. Okay, now we should describe a little bit details about these ovule. Let's find out how the ovule looks like. If we enlarge the structure under the microscope, it will seems to look like this. It is a typical anatropous ovule. Anatropous ovule. In different plant, you may find different kind of ovule. It may be of hemitropous, it may be of campylotropous. I have drawn the anatropous. Typically, this kind of ovule are prevalent, more prevalent in angiosperms. Okay. So let's find out the structural details of ovule. This is an enlarged view of typical anatropous ovule. Typically, an ovule consists of two major portions. Ovule or macrosporangium consists of two main portions. At the base, this stalk-like structure is known as funicle. Funicle. And this entire solen structure is known as body. This is the body of the ovule. Generally, a special kind of cell, parenchymatous cell, consists the body, and these cells are collectively known as nucellus. So by the nucellus cells, this entire body of the ovules are constructed and they are made up of parenchymatous cell. Okay, let's find out its more structural details. The tip portion of these nucellus cells are known as chalaza, and on the other hand, the pore located opposite to the chalaza is known as micropyle. This pore is very important because through this pore the pollen tubes are known to introduced into the ovule and let the male gametes free inside the ovule. Okay, so this pore known as micropyle is very important for the entry of pollen tube. This structure is known as embryo sac. Embryo sac. or it is also known as the female gametophyte. This structure embryo sac within the ovule actually consists the egg cell or female gamete. Okay, so this structure embryo sac is very important within the ovule. So this is how a typical ovule looks like. Now we have to find out the structural details, the formation pattern of this embryo sac, how and where the female gametes are actually reside inside this embryo sac. Now we will discuss about the formation and maturation of embryo sac inside an ovule. We can all remember that this embryo sac or female gametophyte is very important as it houses the female gamete. So come, let's try to understand how any embryo sac or female gametophyte start to develop inside an ovule. Initially, at the developing phase, there remains a large single diploid cell known as megaspore mother cell. So this cell is known as MMC, stands for megaspore mother cell. It is a diploid cell. Then this MMC undergo meiotic division. By meiosis, it forms 4 haploid daughter cells. Those are known as megaspore. So what happened to this MMC? It undergoes meiosis division and form 4 haploid daughter cells. These are known as megaspore, and these are haploid cells. Now out of these 4 haploid daughter cells, any 3 will randomly degenerate. It's a very interesting process in each and every angiosperm flowering plants. Any 3 of these megaspore will degenerate randomly. So imagine these 3 cells get degenerated. Now what will happen? This remaining single megaspore cell will then start to grow. So let's find it out. Imagine these 3 daughter cells get degenerated and the remaining megaspore cell will now grow in size. Now what will happen? This haploid megaspore will start mitotic division. Only its nucleus will undergo 3 rounds of mitotic division, not its cytoplasm. So this mitotic division will proceed without cytokinesis. Let's check it out. Imagine 2 nucleus will form from this existing nucleus after the first round of mitotic division. So these are the 2 nucleus formed from this single nucleus. Now after second round of mitotic division, it will produce 4 nucleus, and at the end of the third round of mitosis, it will form total 8 nucleus. So the nucleus remaining inside this ovule will undergo 3 set of mitotic division and produce 8 nuclei without the division of cytoplasm. So with time and development, these 8 nuclei then start to orient themselves inside the embryo sac or inside these megaspore cells. Now let's find out the orientation of nucleus inside the embryo sac in a separate diagram. So let's find out its orientation in this portion of the diagram. So this is the embryo sac. Now what will happen? Out of these 8 nuclei, any 3 nuclei will start to move towards the chalazal end, and with the formation of cell wall, they will form the antipodal cells. So any 3 nuclei out of these 8 nuclei will start to move towards the chalazal end. This is the chalazal end of the ovule, okay, and they form the antipodal cells with the formation of surrounding cell wall. These are antipodal cells, right? From the remaining 6 nuclei, another 3 set of nuclei will start to move towards the micropylar end. As we all know that this is the micropyle. So another 3 nuclei will start to move towards the micropylar end, and along with their formation of cell wall, they will form the egg apparatus. So these 3 cells will form the egg apparatus. This egg apparatus is very, very vital, because in this egg apparatus, the centrally located cell, this centrally located haploid cell is now known as egg cell. This is the female gamete, okay? This is the egg cell, and the cells located on the both side of the egg cells are known as synergids. So together these 2 types of cell form the egg apparatus. This is the egg apparatus. So clear now? So 3 nuclei form the antipodal cell by moving towards the chalazal end, and another 3 nuclei form the egg apparatus by moving towards the micropylar end. Now the remaining 2 nuclei will align themselves near the middle of the embryo sac, and after few time, in a full grown matured embryo sac, these 2 haploid nuclei will fuse together to form the polar nuclei. So what will happen? These 2 haploid nuclei now get fused with each other and form the polar nuclei. So this is the polar nuclei. This is diploid in nature. All other cells, the cells of antipodal cells or the cells of egg apparatus, are haploid in nature. These cells all are haploid in nature. Only the polar nuclei are diploid in nature. So it is the polar nuclei and the egg cell out of the egg apparatus are 2 important cells. Those take part in the double fertilization that will be discussed in our next class.